So it's my pleasure to introduce next speaker, Professor Jenny Ting. So Dr. Ting is a William Rand Cannon Professor in the Department of Microbiology and Immunology at University of North Carolina. She's also Director of Center for Translational Immunology and the co-director at the Institute of Inflammatory Disease at the UNC. Dr. Ting is internationally highly regarded as being the first to describe a large family of now very famous intracellular sensor of innate immunity, now known as the NAR. It's the NOT-like receptor family. And as a leading investigator in the studies of these very important family members. So her work on this family indeed is transformative and has shaped our current view on the mechanism of innate immunity, cancer, and inflammation. She has published close to 300 original articles and review papers, but more remarkable is 50 or maybe 60, over 50 or 60 of these were published in Science, Nature Immunology, Cell Immunity, and the like. And obviously she has received numerous awards and honors. Just mention one more recent and more notable one. She was invited to deliver a lecture at Nobel Forum in Stockholm in 2014. So please join me in welcoming Dr. Ting.
Thank you so much, Futong, and thank you for your support and the support of all the Yuanshi. It's a great inspiration to follow the talk of Zhang Yuanshi, where she has done tremendous work in terms of translational medicine that most of us can only envy. And I also like to thank, again, I feel like part of Academia Sinica, even though I only spent 6 years of my life in Taiwan, but I feel like this is really my home. And every time I come home, I love the people and I love the food. So and I'm expecting to gain some weight. I also want to apologize because several of us agreed to go to a meeting in Taichung, so I'll be missing the afternoon talks. I apologize to the speakers. I also want to apologize because I just got off the plane from the US about 4 hours ago. So if I start to become incoherent, that's the reason. They always say lack of sleep is equal to 4 drinks of alcohol. So if I start tumbling, that's the reason. It's not because I've been drinking. Oops, so. This is not. So this is actually my University of North Carolina. It's a really beautiful place, and this is our new cancer hospital. So what I'd like to tell you today is about innate immunity and some of these receptors that we have found, and others have found as well, and their role in a lot of diseases. So to think about the balance, I think as Chinese, we always like to think about balances that we have to have, whether in health and life, etc. So in essence, in health, we're really talking about the balance between pro-inflammatory and anti-inflammatory factors. And so if you have a lot of this, you may get into diseases. You have too much of this, you might not be able to fight diseases. But you can extend this into tumors as well. So there are factors, obviously, that will promote tumor growth, others that are suppress tumor growth. So in many ways, we are at the seesaw of trying to determine what is health and what is disease. So we came into this whole aspect because my lab spent years looking at this group of proteins called NOD-like receptors. We initially actually named this, and officially it's named as NBDLR for nucleotide binding proteins with leucine-rich repeat, because we really don't know the function of all of these genes. But just to put into this context, for years, innate immunity was always the weaker sister in immunology, and everybody studied adaptive immunity, until, of course, the discovery of many of these receptors we now know innate immunity is mediated by very specific receptors that signal in very specific ways. And that's our contribution in terms of coming up with this family. So when we were looking at this, we primarily were looking at this protein, C2TA, which was discovered by Victor Steinle. And this is a remarkable protein in that it's a transcription factor. It's a master regulator. And when you put it into any cell, they'll start to express these proteins called class 2 major histocompatibility complex proteins. So they cause tissue rejection, they cause T cell activation, etc. So this is this protein. And we were looking at this protein and realized it has an MBDLR domain, and two people in my lab who deserve great credit, Harden and Lindhoff, decided to search a human genome, and they came up with 22 genes. And at the time, 4 of these are already known. By that I mean there's at least one paper describing these proteins, but the rest were really not known. And so many of these were the ones that we have found. So we at the time named this different names, and finally we came down to the NLR family. And this family has lots of functions. And for this talk, I will only focus on 2 issues. One is this role in inflammasome, which will be clear, and another one is this role as a negative regulator. So the function of this family of protein will be very, very broad. Therefore, the impact is going to be very broad. Like I said, there are 22 family members in human. You don't need to look anymore. You won't discover anymore. Our paper pretty much showed all of them. Unlike TLRs, where for years they start trickling 1 or 2. But in the NLR field, I don't think you need to look. Now, the protein that many people have focused on, and we are not the person who described this, is the Nrp3 protein, first discovered by Hal Hoffman, which he never really got very much credit, as much as he should get. He was a young rheumatologist who was looking at human diseases, and just like Dr. Chang was looking at these links of genetic defects with human diseases, found that this is a regulator of very important cytokines called IL-1 and later IL-18. And the way this is done is these NLR proteins, through many regulatory processes, eventually come up with this very cool looking, almost like a spaceship. And what it is a composition of proteins that forms a fibril. And it nucleates and then starts to grow longer and longer. And what it is each one of these has a multimeric NLR protein, here Nrp3, adapter protein, which also is a multimeric called ASC. And these recruit procaspase-1, which is an enzyme. So procaspase-1 is, after they're assembled into this big structure, procaspase-1 is cleaved into caspase-1, which then cleaves the immature IL-1 and IL-18 into their mature form. So this becomes the seminal part of this maturation process. So this NLR inflammasome has been implicated in many, many things, including metabolic diseases, infectious diseases, viral diseases, fungus, bacteria, autoimmune diseases, etc. But at the time when we were doing this, it was not really linked to cancer. So this is about 6 years ago that we, in this highly competitive field, 3 groups published at the same time. So we all published the paper within 2 months of each other. And what we found was that Nrp3 strangely protects against ulcerative colitis in one model system. And I want to be very clear, different models are very different in terms of revealing the functions of proteins. And then in this specific model, which is using DSS, essentially like a detergent, to induce a colitis-like phenotype. And so colitis is found in both the US and in other countries. And it's actually a risk for colon cancer. So after 10 years, 5 to 10% will have colon cancer. After 30 years, 15 to 40% of people with ulcerative colitis would have colon cancer. So the model, again, is you have colitis induced by this DSS, multiple treatments. You can also inject in the beginning a mutagen, which then, because of the combination of inflammation and mutagenesis, at the end what you get are these polyps. So they're adenoma polyps that you can also see in patients. So this is a model system that's been used very frequently to look at if a gene is important in colon cancer or colitis. And what we found was this paper published in JEM showing if you treat wild type with AOMDSS, you start to see some polyps. But if you treat Nrp3 knockouts, you see much more bumpiness in the colon. This is actually endoscopy, pretty much what we do when we have a colonoscopy, except you use much smaller instruments for mice. So very tiny little things you poke into the mice. And then you see these polyp formation. You can count them. And here we see that if you don't have this gene, there are higher amounts of polyps and the sizes are bigger. And so basically, at the end, what many of us have found is that this gene, remember, causes IL-18 to be produced. And IL-18, in this particular case, is a good product in that it prevents pro-inflammatory cytokines and PSAT in a model of colon cancer. So we think that's why this knockout actually have more polyp formation. Again, in different cancers, this protein will have different roles. And even though it's not published, we have unpublished data to indicate that. So I just want to be a little cautious. So then writing a grant. And of course, in the inflammasome family, there are many, many different inflammasome proteins. Some are important for, like Nrp3, for bacteria, and a lot of other stimulus. But this protein, called absent in melanoma, or AIM2 inflammasome, is important because it is a direct receptor of DNA. So this is a really nice mechanism. So DNA binds to AIM2, and then it changes its conformation, associated with this adapter I told you earlier in procaspase-1. And this forms this very spaceship-like structure, and then it gives out caspase-1, which cleaves pro-IL-1, IL-18 into their mature form. So we just thought, when I was writing the grant in terms of completeness, I just put this in there and said, we should also look at AIM2, because I'm sure what's going to happen is that AIM2 is going to be protective in colitis and colon cancer in both of these diseases, just like everything else. There's nothing unique about this. So just to give you a summary of AIM2, AIM2 is a DNA receptor. It forms an inflammasome. However, others have found that the lack of AIM2 in tumors can correlate with reduced survival in colon cancer patients. And if you put back AIM2, the colon cancer cells, it reduces their growth. So maybe there are functions of AIM2 that's different than this spaceship formation of caspase-1. So it's always suspicious. But we actually didn't think about this. So Justin Wilson started doing experiments, and he found, unlike all the Nrp3 knockout, which people always think they're exactly the same, what we found is that AIM2 knockout, they're no different than wild-type mice. There's no difference here. This is looking at weight change, percent survival, no change. We look at colon length. If the colon is inflamed, it actually shrinks. There's no change. And pretty much everything else, there's no change. So the reviewers made us go through torture and made us do 3 or 4 models, and we found no difference. So we said, there's no difference, we can't find it. But what was really interesting is that we start to look at IL-1 and IL-18. Remember, the inflammasome, the end product is IL-1 and IL-18. What we found is if we look at wild-type colon versus AIM2 colon, there's no statistical difference between these two in terms of IL-1 and IL-18. As a positive control, we have ASC, and this knockout mouse have very little IL-1 and IL-18. So again, this told us that the AIM2 function in this model is probably different than its typical function as an inflammasome. So then we look at the histology, and the histology is really drastically different if you look at AIM2 knockout and an ASC knockout. ASC, remember I said, in this knockout, you don't have IL-18, so you have lots of STAT3 activation. You get lots of inflammatory responses indicated here with all these cells. And here, the AIM2 knockout, they don't look that much different from a wild type that's been subjected to this whole scheme of treatments. So again, we think AIM2 may be different, has a different function than its typical function in inflammasome, exemplified by the ASC knockout. Then we went forward to look at a colon polyp formation in the AOMDSS model. And here you can see that the AIM2 knockouts have a huge amount of polyps, and this will be indicated here. And this is just stretching out the colon. You can see all these bumps. These are polyps. And in the ASC knockout, they also have some bumps, but not quite as much as this. And this is the wild type. So AIM2 did protect against colon polyps indicated here, but it's higher than ASC, and it has a different reason. So here what we found is if you look at this chart, we're looking at IL-1 again and IL-18 again. This is wild type, this is untreated, so they're not that much different. And in the cancer model, the wild type has increased IL-1. The AIM2, if it's inflammasome deficient, it should have reduced IL-1. Instead, it has slightly increased IL-1, but it's no different than the wild type statistically. And this positive control ASC knockout, because it doesn't have inflammasome, so it doesn't have IL-1 anymore. So again, this distinguishes this strain from this strain, telling us that their function in colon cancer models are going to be different from, than in the IL-1 production. So then we looked for lots of mechanisms that we couldn't find. And eventually what we found was this, and this was an aha moment for Justin, who was doing the work. So this is an AIM2 knockout colon, this is a wild type colon, and AKT is phosphorylated AKT, and this knockout has lots of phosphorylated AKT, as you can see on the Western blot, but these guys don't. So the difference looks like it's this pathway. And again, if we look at this ASC knockout, which everybody thought was exactly the same as this, doesn't have any AKT phosphorylation, indicating the reason these two have increased colon is due to different mechanisms, because so far they don't look the same in any of the things that we looked at. So this one must be going through a different pathway than just typical inflammasome activation. And why do I tell you about AKT? Because it turns out AKT is a great tumor marker. So AKT is mutated or lost in many different tumors. For example, in thyroid carcinoma, 80 to 100% of the tumor have an activated AKT. And AKT is pro-survival, so when you have AKT, the cells can grow, and therefore you have tumor. In gastrointestinal tumor, 30% of human cancers have activated AKT. So this is a very active marker for tumors. So then we wanted to know what is the mechanism by which AIM2 can switch AKT. And the answer turned out to be very complicated. So we did a proteomics analysis to see what is different in the AIM2 knockouts versus wild type. And we found one protein that interacted with AKT, with AIM2, and that is DNAPK. So DNAPK interacts with AIM2, and to make a long story short, what AIM2 does is blocks the function of this protein, so it can no longer phosphorylate AKT. That's why when it's present, it keeps tumors in check. If you take away AIM2, this whole pathway is activated. Then you have tumorigenesis. So again, in terms of DNAPK, it has been linked to many different tumors, including colorectal tumor, and it promotes this oncoprotein stability. So DNAPK has been linked to cancer development, and we think that AIM2 usually keeps this in check. So just to show you some data that we have done to look at tumors itself and the link between AIM2 and AKT, this is a colonoid culture. So this is a 3D culture that was that we worked with the Department of Bioengineering at UNC. And these cultures have a 3D structure indicated here. And we can look at wild type and knockout. So these cells don't have AIM2. And you can look at AKT phosphorylation. And this is without an activator. This is with the activator. So AIM2, in both cases, the cells without AIM2 have increased AKT. So verifying what I was saying. So without this protein, you have increased AKT, which is increased survival and increased tumors. When the tumor cells can survive, you have increased tumors. However, we don't see this in macrophages at all, indicating that the impact is really on the tumor. The impact of AKT is really on the tumor. And then I don't want to tell you about this, but we also did these torturous experiments called bone marrow transplant that only immunologists can think of to torture each other. And we did that for the paper, but nobody else really cares. So I won't tell you about it. And then we wanted to know, because this model system is not really that great, the AOMDSS, there are problems with it. It doesn't always mimic what happens in cancer patients. So we went to the second model, which is spontaneous model of colon cancer. And the spontaneous genetic model is always the highest level of proof that something is important for cancer. So we put these, this is a human mutation that we put into mouse, APC-min. This is linked to many types of cancers, including colon cancer in humans, this APC-min. So we put the APC-min mouse and bred it onto our AIM2 knockouts. So here's the APC-min. You can see tumors. There's not a whole lot. And we put it on the AIM2 knockout. Now we see quite a few tumors indicated here. So this, again, if you have this gene, it protects against the presence of colon cancer. And and here's the real counting of the macroscopic polyps. And again, in the AIM2 tumors, we have more AKT. So then we thought other people have already shown that 30 to 40% of cancers actually misses AIM2. And that's because that's attributes to its name. The name of AIM2 is really absence in melanoma, right? So it's actually very low in some melanomas, okay? And when something is low in melanoma, you always think its presence would protect against that cancer, okay? So AIM2, we think, is a tumor suppressor. Its presence suppresses tumor. It's exactly what we need for health. So then we wondered, with all the AIM2 defective tumors, can we think about a treatment? So remember I linked AKT to AIM2. That is, AIM2 prevents AKT phosphorylation. So in the knockout, you would have heightened AKT phosphorylation. So then we asked, can we use the AKT inhibitor and can we prevent tumor in these AIM2 knockouts? And that is indeed what we saw. So this is AIM2 knockout. You see lots of little polyps here, 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 here. And if you treat this with the AKT inhibitor, now you see very few polyps. And this is the quantitation of the data. This is how many polyps you see without treatment. And this is AIM2 knockout, where we treated it with the AKT inhibitor, and the tumor really dropped. So we think this is really great because now we can go to the clinic, look for AIM2 deficient tumors, and think about this therapeutic. So if anybody in Taiwan is interested, we'll be delighted to collaborate with you. So to just conclude for this part, we think AIM2 usually holds this in check, holds this back, so you don't have AKT phosphorylation. If you don't have AIM2, AKT is phosphorylated, but you can take an AKT inhibitor and treat this tumor without AIM2, and you can really reduce tumor formation. So we think this is potential therapy for treating tumors with reduced AIM2 expression. And that is exactly what people are talking about nowadays, about personalized medicine. That is, if you can get a group of people, you can understand what is their genetic defect, you can actually figure out a way to treat their cancer. So then I want to touch on another topic. I told you about this AIM2 unexpectedly is a tumor suppressor. Now I will tell you some more about another group of NLR proteins. So there's a number of NLR proteins that turn out that they have an immune suppressive function in terms of inflammatory response. And I'll tell you about one of them. And our lab is responsible for cloning many of these proteins. So several groups, including the Laboratory of Theramala at St. Jude's and Rongfu Wang, have shown that several of the NLR proteins turn out to be suppressors of inflammatory response. So this is just giving you an example. So this is very immune-centric view of NF-kappa B, which is a major factor in almost every field. But in immunology, we think of it as an activator downstream, for example, of the toll-like receptor, activates to MyD88, activates IRAK1, TRAP6. Then you have IKK, and you have the whole IK beta phosphorylation leading to gene transcription. And then you have a non-canonical pathway here. But regardless, several of these NLRs have been found to be inhibitors of these pathways. And I will tell you about one of those. So this is a study by serendipity. So we were the first group to describe a protein called NLRX1 in 2008. But another group, Samit Chanda, at the Burnham Stanford in San Diego, was looking for proteins that might inhibit our host response to HIV. Because if you can find those proteins, you might be able to manipulate them so we can increase our host response to HIV. And what he found was when he did this with a non-biased approach of siRNA, he found this protein called Nrx1. And that's the one we described years ago in terms of being a negative regulator of inflammatory response. So we describe it as a negative regulator of toll-like receptor and Reg-I-like responses. It downregulates NF-κB and IRF3, but you don't need to know the detail, but that is a suppressor of the immune response. There's some conflicting data about this protein. We're still working on it. It may have different functions at different places, but I'll tell you about what we have found recently in collaboration with Sumit Chanda's lab. So Haitao Guo in my lab then decided, Sumit called us about this finding, but Haitao at that point just joined my lab, and he's an HIV person who was just starting the study. So what he did is knock down Nrx1 using siRNA. So you can see expression of Nrx1 normally is this high, and he knocked down Nrx1. And what he found was when he did this, if you look at HIV infection, when you knock down this gene, HIV infection is decreased. So let me just go through the logic of this. So this is a suppressor of immune response. If you take this protein away, you have increased immune response. Therefore, HIV infection went down. So there's several levels of negative thinking here. So what we did is when we knocked down this gene, HIV infection went down. And we can do this with a single round HIV virus, or we can use a real HIV virus, HIV-R3A. And we can use different ways of looking at this. And in every case, what we found is that the immune response, such as interferon beta, if you knock down this gene, indicated here by shRNA, interferon goes up. And again, interferon is one of the best antiviral proteins that our body produces. And again, here, if you knock down this gene, interferon goes up. So again, this is a suppressor of the immune response. You take it away, all the immune responses go up, and this is illustrated here where we're looking at a whole bunch of cytokines that are antiviral, and all of them went down in the black bar when you take away Nrx1. And this is present in macrophages, mouse cells, MAPS, etc. So then we wanted to know at what stage is Nrx1 inhibiting interferon. And so this is an experiment that is typically what HIV people do, and it's a little bit hard to explain. So what you do is you can take a RT reverse transcriptase inhibitor, and what this does, this inhibitor, you don't know, again, don't have to know the details, but this takes away the presence of the DNA part of HIV. So you do away with the cDNA component of HIV because it's a reverse transcriptase inhibitor. This inhibitor is an integrase inhibitor, right here. So the integrase inhibitor does not affect HIV cDNA. So the cDNA is already made. It just cannot go into the nucleus and integrate. Okay, so you're stopping at two different points. And a couple of years ago, it was found that this is the phase, that the cDNA phase is the part of the HIV that induces interferon. It's the DNA phase. So we wanted to see if Nrx1 is impacting the cDNA phase or post-cDNA phase, and this is shown here. So if you take HIV, you get interferon, you use an Nrx1 deficient cell line, and interferon is increased. Remember, this is a suppressor of interferon, so you take it away, interferon is increased. Here you have integrase. So cDNA is already made, but it's not integrated. And it doesn't matter because the cDNA is the one that induces interferon. So with Nrx1, this is the amount of interferon you see. Without, this is the amount you see. And if we have no reverse transcriptase, we don't have the cDNA phase, we still have a little bit of interferon, but you take away Nrx1, it doesn't make any difference. So this suggests that Nrx1 is changing the cDNA phase of HIV infection. So what is the DNA recognition that induces interferon? So we wondered if Nrx1 then will integrate with this part. And this is major discoveries made in the last 2 years, primarily by a number of labs, but most by James Chen's lab. And what he figured out is double-strand DNA, when it gets into a cell, or when it's produced by the cell, and it's in the cytosol, it's recognized by this enzyme called cGAS, which then produces this dicyclic nucleotide, which then binds to this protein called STING, which then activates TBK1, which then activates Nrp3 and induces interferon production. So this whole step-by-step dominoes effect is seen here. So cDNA starts, and so this DNA can be from HIV, inducing this whole step. And what we found earlier is that NRX1 interferes with probably this pathway, because this pathway is the major DNA-induced interferon pathway. So we wanted to see if NRX1 might interfere with any of these steps. And to make a long story short, what we found is NRX1 inhibits this step called STING and TBK1, which is very critical for downstream signaling. So this is the first experiment to indicate it inhibits STING, and this is an artificial experiment, which we have empty vector, and we see interferon promoter being induced very well. You add NRX1, it's reduced. Then the second step is we wanted to see if NRX1 can interact with STING, and this is all endogenous protein, and it did interact with STING. Then we wanted to see if they come together, and we did a co-merging of the two, and STING and NRX1 staining. One is green, one is yellow. If they come together, they become yellow. One is red, one is green. They come together, it's yellow. And indeed, we are able to see all these yellow. And this staining suggests that they're coming together, just like this experiment suggests they're coming together. So what we think is going on is NRX1 may be binding STING, so it can't work anymore and can't activate the downstream signals. And so that's what we wanted to know, and that is indeed what we saw. So this is looking at STING by itself, and STING normally can pull down this protein, which is critical for the downstream interferon response, and it did pull it down. But if we titrate in NRX1, these two proteins start to interact less and less and less. So the conclusion is that the presence of NRX1 blocks the interaction of these two, such that the interferon pathway cannot occur. So to give you the model then, what we think is going on is HIV, which is a single-strand RNA virus, has to go through a phase to become DNA. And of course, that's what David Baltimore found, which gave him the Nobel Prize. So this double-strand DNA is the one that then goes through nuclear import and goes into the cell. But the DNA here induces this whole pathway of interferon, including STING and TBK, to give you interferon production. And NRX1 is the one that blocks these two from functioning. So while we were doing all this, another group headed by Dan Baruch and mostly Sakali was kind enough to call us before his paper was published and said, we have this data, and we wonder how you can interpret this, which is in primates that are infected with HIV, right away what he found is there's an inverse relationship between NRX1 and all the interferon-inducible proteins. So whenever NRX1 is high in his non-human primates, what they found is that these interferon genes are low and vice versa. And that's exactly what we are saying, that NRX1 is a suppressor of interferon response. So his data in high-throughput screening of non-human primates agree exactly with our very mechanistic work trying to figure out what it is doing. Now, I just want to finish up by saying, so why do we want negative regulators of immunity? You would think that if you have strong immunity, you can get rid of HIV, and that's what you really need, right? And so I just show you some reason why we want these negative regulators of immunity. And this just shows you human studies that we have done by looking at the Oncomine database, hundreds of colon cancers. And this is normal controls. And NRX1 expression is lowered in colon cancer. Then in addition to that, we also collaborated with the 4th Military University in China. And from primary tissues, what we saw is, again, normal tissue compared to colorectal cancers there's a reduced expression of NRX1 in colorectal cancer. So then we put these mice, NRX1 knockouts, into the same model that I was just telling you. This is the AOM-DSS model, and the knockout has increased cancer. Remember, this is a suppressor of immune response, and so here we got lots of inflammatory response, and it's known the more inflammation you have at the site of tumor, the worse the tumor becomes. And that's exactly what we're seeing here. We have more tumors here. We also went to the AOM-DSS, to the APC-MIM model, which is a spontaneous cancer model. This is how much you get with the APC-MIM per se in terms of survival. But if you put it on an X1 model, they really accelerate, and these mice do terribly. So without this NRX1 to protect the mice, in the absence of NRX1, they really survive poorly. They have increased tumors, and the tumor volume is increased. And what's really interesting was then we tried to figure out what is wrong with the tumor, and we found out that these NRX1 knockout tumors have increased cytokines such as IL-6. And so then we wanted to know if it has increased IL-6, can we treat these animals with anti-IL-6, which is really used for treating rheumatoid arthritis, not to treat tumors, and can we get rid of these tumors? And that is exactly what we saw. So here, this is the amount of tumor in an NRX1 knockout that also have the APC mutation. So you have lots of tumors. You treat these animals with IL-6 inhibitor, and you reduce the tumor significantly. And here again is the number of polyps without anti-IL-6, with anti-IL-6. Here's the signaling pathway without IL-6, with IL-6. So we think this is, again, another possibility of personalized treatment. So here we found that NRX1 expression in the colon crypt serves as a tumor suppressor. So when this is present, it suppresses tumors, and we found that this protein is reduced in colon cancer patients. So this is tumor suppressor. This keeps on going forward for some reason. So if we target this with anti-IL-6 therapy, we can reduce polyps, and we wanted to know if this is potentially a way for personalized cancer treatment. And now we also know NRX1, the presence of this protein can reduce COPD, for example, and can reduce multiple sclerosis. So again, back to this. We know health is a balance between pro-factors and anti-factors, and I've shown you two of these, which are suppressors of tumors as well as inflammation. And so I think ultimately the summary is that we long know that this is an issue. So Chinese people always say, we figured out this thousands of years ago, and that's kind of true. Because the yin-yang model, we need yin and we need yang, and we have a balance, and that's exactly what we're trying to say, is that in immunology or in tumor cancer research, the balance between the two is really critical. So just to acknowledge the really talented people and collaborators, Coy was the first person who did the AOM-DSS model in our lab. He's now a faculty at Virginia Polytech. Justin, Alex, Leo did the AIM-2 studies. Alicia, Aga, Rong did all the studies with the NRX1, and Rong is now a faculty in China. Haitao and Meng did the study with the HIV. These are our collaborators at UNC, and these are our collaborators elsewhere, including our collaborators in China. And these are our funding agencies. I just want to thank you so much for your attention and time, and it's a great honor to give this talk.
Thank you very much for your inspiring presentation. So, question is.
Very nice talk. I have two questions regarding the AIM-2.
Yes.
So the first one is, have you analyzed the cancer patients to see whether AKT is really activated in cancer patients?
Yeah, that's what we're trying to do right now.
Okay.
Like I said, we would welcome any clinical collaborators if we can start looking at things like that. We have a group in China, and the Fourth Military University has one of the largest collection of gastrointestinal cancer. So we're working with them on that.
Okay, my second question is about the double-strand DNA for M2.
Yes.
What is the source of the double-strand DNA for M2?
Yes. That's an excellent question. So this is a area of debate. How do you get cytosolic DNA? And there's at least 2 studies now. One is a study with HSV, which showed that the virus goes through a stage where you can detect DNA in the cytosol. Because we always think about DNA as being
Right, but
nucleus.
it's understandable for the NLRX1, right?
Right.
But for M2, we don't have any virus infection.
No,
for
M2 can respond to DNA viruses too.
I know, but for the colon cancer,
Yes,
story,
so comes to my second point. So a collaborator of ours, Aziz Sanjar, who just won the Nobel Prize last year, he showed for years he had these papers that nobody believed. So he could only get them in a really good journal, but not your Cell Science Nature, like JBC, right? And where you have the best papers, to be honest, is in JBC. And he showed that when you have DNA damage, you have these small cytosolic DNA that's in the cytosol. And I hope that now he will get a lot more press because now he has a Nobel Prize, that people will believe him a lot more. But that's what he's found in the JBC paper. So I think we're really working actively to figure out that that's the case. And I think that will be the case.
okay, thank you very much.
Sure.
A great talk.
Thank you.
I have follow-up on the link between N2 and the AKT
Yes,
phosphorylation. And you attributed this to the dysregulation of the DNA PK.
Yes.
But I noticed that in your Western blot, there's also a lot of PTEN function
Yeah.
in the AON
Yeah.
So could that be an alternative
So
mechanism?
the P10 data was not that reproducible. So that's what we were focused on for like 6 months because I was so excited. As you know, P10 is a huge tumor suppressor. And I couldn't really say it's always reproducible. We now have data that if we put AIM2 on a DNA PK, AIM2 knockout on a DNA PK knockout, there's no impact. So that's always your experiment to say something is linked to the other, right? You make a double knockout. And when we did that, we don't see any impact yet. So we think the two are really linked.
I'm also interested in M2. So does M2 inhibition of DNA PK requires the assembly of the oligomeric structure, just like AAC2?
Yes, we don't know at all. So we have one guy who's really interested in looking at this. In our model, it would be completely different. Because AIM2 in the inflammasome is that spaceship-like structure, right? We think AIM2 in DNA PK is probably different.
You know the interaction domain.
We're looking at that right now. So unfortunately, my postdoc is on to other things. So he's like kind of lost interest already. He's on to the microbiome, which I hope I'll be able to talk in a year. But he's kind of hasn't gone back to asking these same questions. So. We did map some localization domains and so forth.
Yeah, maybe one last question.
Yeah, my question regarding subcellular localization of STING and NLRX.
Yes.
So this localization is in mitochondrial or in ER.
Yeah,
and
so-
what mechanism will cause this?
Yeah, so in our paper, the reviewers asked that exact same point. So we had to go through this torturous MAM, the mitochondria-associated membrane, which is a fusion between mitochondria and ER. And we got those preps from a collaborator, and we were able to detect associations. So many proteins, when they're found in the ER, they're also found on MAMs. And proteins that are found in mitochondria are also found on MAMs. So I think when you look at it, it's really a continuous continuum. So that may be where they're interacting.
So you prefer the NLX regular synapse between the mitochondria and ER?
Yeah, we have not really done exhaustive studies. The NLR localization is a little bit complicated. So when we are looking at cell lines, we have reproduces over and over, and actually we asked 2 other labs to reproduce it for us. One is James Chen, and one is the laboratory upstairs from us. They're an expert in p53 mitochondria, and they always think it's the outer membrane protein. When we start to look at non-transformed cells, we start to see a different assay. When we start to see viral infected cells, we see a different localization. So it's a nightmare, but our feeling is we really have to figure it out where it is. So it is possible it moves. But when you think about many proteins now, they actually do move. Like STING was found in the ER. So everybody says STING is in the ER. It turns out STING is also in MAMS.
Okay, so I'm sure there's still a lot of questions, but we don't want our new president to wait. Jenny will be here for the whole week, so maybe we can try to catch her when she's here for the convocation. So let's thank her.